I don't know if you can hear me. So welcome to this special lecture of the ELIXIR-CZ structure, which will be given by our colleague Pačes from Institute of Molecular Genetics. You probably noticed that 2 years ago it was the corona pandemic, and so we probably noticed that 2 years ago it was a corona pandemic, and from this institute you probably noticed that few of the representatives were very much active in the public sector. But there is also kind of the research part of it, and this is the genomics. And many people from the research laboratories actually gathered all the sequences of the coronavirus, so the genomics was a really important part of the whole cure of or at least fight against the coronavirus. We are lucky that Czech Republic has a few esteemed genomists, whatever. And Honza and his colleague were active, and actually they started something like an activity which is purely academic as a coronavirus genomics, and COG, you probably noticed that acronym. And they are responsible for the kind of the standardization of the genomics approach towards the coronavirus. Better than to actually talk more about it, I would like to say that Honza is one of the builder of the bioinformatics in the Czech Republic. He is also one of that, I think, basic people behind the ELIXIR and the part of the Institute of Molecular Genetics. So Honza, the floor is yours.
Okay, thank you, and thank you all that you are willing to spend such a beautiful morning here with me. I'm going to show you what we learned during these 2 days, 2 years, what the pandemic was really about and what we can maybe expect in the near future. And the thing is that virology was not really big news at that time, and the reason is that when you would like to observe, to find new virus, that's not easy. First you have to find an animal, you have to understand how the animal behaves, then you have to find the animal when it is ill, then you have to find the reason why it is ill, and then finally you can find the virus. So at the beginning, viruses, beginning of genomics, like 50 years ago, viruses were really great tool because they were small and relatively easy to work with. But after the modern technology, everyone from the genomics moved to others, to the normal life, not to the viruses. Now, we are in a little bit different situation because of that particular pandemic, and believe me that everyone, including me, was going through this nice Dunning-Kruger effect. So, what is that coronavirus? Coronaviruses are single-strand RNA plus viruses. It means that they have their genome encoded in RNA, not in a DNA. And from the virological point of view, these are really big RNA viruses. Its length is around 30 kb. And so far we knew 2 years ago, we knew 6, and now we know 7 human coronaviruses of 2, genus alpha and beta, and those first 4 with abbreviation ACOV and whatever, these are normal coronaviruses which every one of us usually get infected during the winter. Then there are 3 which are much more dangerous. The worst of them is a MERS, which is a coronavirus which jumps to the human population from camels, and then 2 SARS-CoV viruses. The SARS is from severe acute respiratory syndrome, and these are those who are now making our life difficult. This is how the coronavirus looks like under electron microscope, and you see the reason why they are called coronaviruses, because they have such a special corona around them. This is another picture showing the cell, which is producing a really huge number of viral particles, and what you can see that the main focus of coronavirus is to produce as many particles as possible, and it does not care much about the quality of the particle. It just produces. It's like crazy with a lot of errors. They are different sizes, not complete, not infectious, but also infectious as well. And this is the model. You probably saw you all saw this model. There are some really important features. This is a virus with gray. This is normal membrane, and from membrane there are 3 proteins going out of the virus, and this is the most important one. It's called spike protein. It's a trimer, and this is the protein which allows the virus to infect other cells. And also this is the only thing which is actually visible for our immune system. So from immunological point of view, these are regions where our antibodies are directed against. This is from one of the first articles about the mechanism how SARS-CoV-2 enters the cell. What is important that there are 2 ways how the coronavirus can enter the cell. One is, this is the main. At that time, it was not clear which one is the major one. So this is the major one, that the spike protein binds to Ac2 receptor, then it cuts out by the protease, and then the membranes fuse, and the RNA is entering the cell, and then all the machinery starts. There is another way how it can enter the cell, where after binding to Ac2 receptor, it goes into the endosome, and then it's processed out of the endosomes. And this is the way how it's then processed, the genetic information from the virus, how it is processed in the cell. What is important is this Ac2 receptor. This is the key protein which allows the virus to recognize the target cell. And that's the reason why it makes so complicated situation, the virus, because Ac2 receptor actually is presented on surface of many, many cells. Of course, these are the lungs and bronchus, that's clear. Of course, it's a respiratory virus. It goes to intestine, well, as usually these are the same epithelial cells, but it goes to really number, it goes to the livers, to kidneys, to pancreas. In men, it attacks testicles, seminary vessels. In women, it goes to the placenta, so it's very dangerous for pregnant women. And that's the reason why there is a huge spread of different symptoms you can get depending on what cells actually were attacked by the coronavirus. Fine. From the genetic point of view, it's relatively easy. It has 10 genes, but when you look, and this is the spike gene, which is really the key, and most of the research is directed against the spike protein. But of course, as a viruses are working with their genetic information in a very special way. So if you look at the proteins and transcripts which can be derived from those 10 genes, you see that it's quite complex. It creates a huge polyproteins. It has an alternative alteration of the reading. So there's a lot of really nice things going on there, and it produces much more proteins than actually it has genes. But fortunately, we are in a new era, and you here at the Institute of Organic Chemistry and Biochemistry know that now we can easily create 3D structure. So few months after the genetic code or genetic information of that virus was released, it was easy to get 3D structures of all proteins which are part of the virus. And these are this is the most important one as a spike protein. This is the red part. It's present as a trimer, and it has several features. And on top of this, this is the region when it actually binds to the Ac2 receptor, and it can have two stoichiometry or two conditions. One is closed. This is, for example, the blue one, and the other one is opened, and it has to be open to bind to the virus. I have also the 3D printed model where you can see it. Here you do not see the transmembrane part of the protein. Here is the monomer which is in open position, and this is Ac2 receptor, and this is the way how they binds together, and then everything starts. Okay. Also, they printed me with such a strange plastic that it glows in the dark. It's some creepy green, so you can play with it. It's nice thing. And it's really precise on angstrom level or something like that. Fine. So that's the coronavirus, it's a special SARS-CoV-2. Now let's move to the mutations and variations, because that's what everyone is interested in. So as a normal RNA virus, it mutates during the time. Coronaviruses usually mutates in much slower rate than other RNA viruses because they have a special enzyme, exonuclease, which helps them to repair during the multiplication. And the speed roughly is 25 mutations, substitutions per year. It's roughly because what you can see here, when there is a new variant, usually there is a little jump. This is the dark blue, it's variant alpha, and in red it's variant Omicron, and you see that there is a very often such a small jump. There is usually the variant which starts to spread have more mutations than would be expected, which is really nice to monitor. Now the whole pandemic had 3 stages so far. First stage, and I will document it on the mutations, was during year 2020, where this is a genome of the virus with the genes you see here, and here is are mutations on a amino acid level. So, and it's entropy, not events. And you see that at the beginning, the mutations were everywhere roughly equal. Those peaks, these are mostly because it's entropy. These are from the regions with high entropy. And of course, that was the reason was that at that time, the virus had free space for expanding, and there was no competition between the different strains of viruses. So each virus can create any mutation, and because one mutation usually does not change the proteins really much, so the differences between these all variants at the beginning, was hundreds of them, were not so big, and because the space was infinite for the virus quickly spread through the whole world. Then we become a little bit nervous about that, and we closed the borders. So at the beginning, it was really luck, pure luck, and random events. And in each country, then you got a set of variants randomly chosen for all the possible ways, and they start to grow in different countries because then the travel stops between the countries. Then in 2021, there was a possibility to get a vaccine, and also a lot of people were already infected, and as a population we slowly built the immune defense against the virus. And at that time there was not enough free place for the virus to expand, and different strains start to compete between each other. And this is what we see on the second part, where there are now several mutations which really change the behavior of the virus and allows that particular variant to be faster or outgrow the other variants. There was the second stage of the evolution of the virus and its human host, new human host. And now we are in the third stage, where you see that definitely the mutations we are now observing, of course the mutations are still random, that sometimes if you sequence any sample you can get mutations everywhere, but they are not spreading anymore. Only those which really change the virus are spreading, those which are changing the behavior. You see, of course, that most of them are on S protein, because that's the most important with interaction in its host. And also, now the reason why we see this secondary mutations is that at the beginning you were not able, the evolution or mutation, there was not enough time to do any amino acid change, because when you have time only for one mutations, you cannot do from any amino acids any other amino acids. So you have to have 2 mutations on that particular position, or 3 even, and to be able to have 2 mutations first, the first mutation have to be fixed, okay? But now, number of mutations are already fixed in the population. Virus is a little bit different than what was at the beginning. And now we are seeing the other mutations which are helping the first mutations which were fixed at the first expansion of the virus. And also mutations which were not possible to observe at the beginning of the pandemic. Now, there is quite a big problem with naming convention, because of the epidemiology. People prefer to talk about mutations on an amino acid level, which of course from the genetic point of view make no sense, because mutations is on RNA level, right? But anyway, so we are now talking about the mutations on amino acid level. And this is the scheme how they are named. So it's an S protein and amino acid asparagine N on position 501 was changed to amino acid tyrosine. This is how those are named, but because it's so complicated, many of those important mutations has their own name that virologists can talk about that. So for example, this one is called Nelly, we have EEC, we have POO, and so on. So these are names for the mutations when we are talking about. Some of these mutations are really important. They are changing the virus, and that means that when they happen, that is very likely that they will be fixed in the population. We can see it, for example, in that mutation Nelly, that all those first 3 strains, this is alpha, this is beta, and this is gamma, I think, have the same mutation, but not by origin, but this is a new mutation and new fixation. But some of the mutations are there just because of the chance, and they are there from the beginning. For example, DAUK, D614G, every virus in the Europe has this particular mutation, and the reason is that this is the mutation which actually were first fixed in China, and then it spreads to the world. So that's the change. And of course, I'm going to skip this. And now these were mutations, and if you have a set of mutations which actually changes the virus, then usually you name it a strain or lineage or whatever, and there is no clear convention how to name those strains or variants. And for the coronavirus, there are 2 different naming conventions. One is such in an automatic way when there is a region in the Earth where there is a set of mutations which starts to expand together. So it's called variant. And it's named sequentially. So first is B, then B1, B11, B111, and so on and so on. But because we would not like to have these names really long, so when you have three digits, so that's X is substituted by another letter. So C1 is in fact B1111. Okay, that's the way how it is automatically built. And the good thing on that is that you immediately know what was the origin of each variant. There is another way how to name the variant. This is more virological one. The names of these are like 21L. This is is year. It's a way how we name, for example, influenza. This is a year and then the letter. So it started at 19B, 19A, and then 20, 21, and now this last Omicron is 22 something. Okay. It's quite confusing, and there's a lot of misunderstanding in the public what those are. So then World Health Organization came with an idea that we should name them in more friendly way. So they chose the Greek alphabet, which does not help us much because these are alpha coronaviruses, beta coronaviruses. So the delta coronavirus belongs to beta coronaviruses. Well, it's a mess, but anyway, people start to use it. So each of the clade is then named by, which is important enough that we should keep track, is now named by the Greek alphabet. Everybody knows probably about the variant alpha, and then the delta, which was dominant here in Europe, and now the Omicron. The funny thing is that we expected that Omicron would be named new because that was the next letter in the Greek alphabet, but WHO decided that a new sounds the same like a new virus, so they skipped that particular letter, so new was skipped. But then they came with another problem. The next letter is Xi, which unfortunately is the name of the Chinese president. So they skipped it as well. And so these are two letters which were not assigned, and the last variant is now Omicron. And of course, this not last variant, we will see most of them. I will not talk about the sequencing, but we started the sequences at the early of 2021, because the first year it turns out that the Czech Republic is not organized well enough to do it on a state level, that because of it was horrible. So we created informal CoG-CZ coronavirus genomics group with many people. Usually you are showing the collaborators at the last slide, but I'm not showing it in the last slide because this is a really great group, people who are helping us, and we started the sequencing. Now it is organized on the state level, monitoring the situation in the country, and we are creating. We have web page. We are creating bi-weekly situation reports, which you can download, and you see the trees and variants which are here. And I'm going to show you what's there. So now we know what the coronaviruses are. We know how it is with mutations and variants. And now let's have a look what actually happened. I had to fill this slide of chapter 3 with some picture, and I use for the first time this particular picture, and I realized that it is really important. Do any one of you know what this means? Yeah, okay, so we are now talking. Usually there is only one or two people understanding why this picture is showing there, because we are going to talk about evolution. And in evolution, we do not see everything. We see only what happened. And this is the picture from the Second World War, where there was a battle for Great Britain, and they did not have a lot of airplanes, and they tried to improve the airplanes. So the general's task was what are the parts of the plane which should be improved? And the technicians had to mark down the places where the airplane was broken, and you see that in the red dots. And after that, the technician came to say, okay, now we see we have to improve, for example, ends of the wings, because there was a lot of broken parts here. Fortunately, the statistician came and told them, no, that's not true, because you do not see hits which are here and here, because those planes never returned. Okay, and that's exactly with the evolution. We see only those events which actually were successful during the evolution, but many more events were not successful. So keep that in mind when you are analyzing the viral infections especially. So at the beginning, for the first year, we do not have much data, and there was a big mess from the genetical point of view, because there was hundreds of different variants slowly start to competing. And then came variant alpha, which slowly overgrow everything which was present there. This is a word view. And then another variant delta came and slowly exchanged the variant alpha. That, of course, was not the same in the whole world. For example, here you see Europe, that we had only alpha and then delta and now Omicron. But for example, in South America, where they had a big problem keeping the pandemic down, there was a number of very strange and probably dangerous variants like lambda and there was another one there. And you see that, for example, gamma and alpha never was very strong there. So at the beginning, the different places on Earth had a different pattern. In Africa, you see variant beta was again quite danger. But at the end, when the variant delta came, it overgrow everything everywhere, and now the Omicron is overgrowing everything. There is a common misunderstanding that respiratory viruses during the evolution should become milder and milder, and at the end the virus will be like a normal flu. And the reason why we think that is that these are the respiratory viruses we see, right? But of course, there were very often it happened that the virus was not milder and milder, but much worse and worse, and actually killed that host. In fact, most of the species on Earth during the evolution were extinct because it did not survive its contact with a new parasite. That's probably the most common way how the species can end. And yes, we see those events which actually were successful. And the reason is that when the world was still divided, that maybe the virus infected one village or one city or even one continent. And if it was really bad, then it killed a lot of people. And because of that, it actually stopped there, and another strain of that virus, which developed in another place and was lucky to be milder and milder, then overgrow slowly everything. It was great in the time when we had a separation of different populations in the world, which unfortunately is not the case anymore. We are one big village on the Earth. So it can happen that this virus will become milder and milder, but so far each variant is worse than the variant before. The direction of that is not really in favor for us. Okay, what variants do we have in Czech? This was alpha. Then it switched during the last summer to variant delta and all these subvariants. AY, these are the subvariants of variant delta. And now we see Omicron. This was the Omicron 1, and then Omicron 2, and now 1.1, and now Omicron 2, and other are coming. Of course, we are not, when we are monitoring the virus, we are not monitoring it on a level of the variants, because variant is nothing really defined. You have several mutations, but of course it does not stop there. During its evolution, it has more and more mutations, and it changes slowly, so you have to monitor it on a level of different mutations. This is quite complicated picture, so follow me. This is a spike protein. These are position of known mutations in that spike protein, which are important for defining different variants. And here you see different variants and the key mutations which are present in that variant. Okay, so each column is one variant. And here you see last year, 2021, each week of that year, and by the heat plot you see what variant was present in that week in our samples. And at the beginning you see those mutations which are part of the alpha variant. So at the beginning there was alpha variant, and then during the summer, alpha variant was exchanged by the delta variant. And of course at the beginning there were other variants as well, and delta variant at the beginning was only one delta variant. But if you look in other positions or other regions of the genome, the reason why it is here, it's a spike protein, and those mutations were so important, then you see that the diversity was much complex at that time. When you see the change of the delta, these are mainly delta, this was alpha at the beginning. So you see that there were variants which tries to grow at the beginning, but very soon were overgrown by the other variant. It was like AY4, AY43, AY9. So there is a really lot of going on when you are monitoring that. It's different in different countries. It goes through the whole world. And of course, the travel makes the situation quite complicated from the epidemiological point of view. So you can play with mutation, observe the new mutations which are starting to overgrow in some particular region, and think about what that mutation is important for. And why we know that some mutations are important, because we have the 3D structure and we can look at that and really understand why the mutation on that particular position changes the behavior of the virus. So this is a mutation Nelly, and it was first spread through the lineage B117. This is the alpha lineage. And when you see, this is the position where it is, and it's directly on the contact between the spike protein and Ac2 receptor, and immediately you can do experiment and to mutate that particular position and see what all changes, and that position will change behavior of the virus. It binds better to the Ac2 receptor or worse. And of course, then you see a number of other mutations which we do not know what they are doing. They are in the middle of the spike protein or other somewhere else, and actually there are there is no many antibodies binding here. But what we see is a number of different lineages where these particular mutations happened and was fixed. So we know that it's probably really important for the virus, because it's fixed repeatedly on different positions. Okay, and for the epidemiology, for the healthcare, it's really important, because if you look at the spike protein, you can see that you cannot create, or our immune system cannot create antibody against all positions. The reason is that maybe this position looks like other protein in our body, and this antibody is negatively selected in thymus, or that that particular part of the protein is coated by glycoproteins, or that part of the protein is just too smooth and the antibody cannot find some special region which can fix on. So in fact, what we are doing, our immune system is doing, we are creating just probably 7 types of antibodies only. Okay, and these are the regions. This is a antibody type 1, type 2, type 3. Different authors have different numbers of those regions, but let's say several different regions. And of course, when there is a mutation in that particular region, it can change the physical property of the protein, and then it can bind better to Ac2 receptor, but also that antibody which we had against that particular region will not be working anymore. And that we see in that monoclonal antibodies treatment, that when you create the monoclonal antibody treatment against the Delta virus, they do not work against Omicron, because Omicron have mutations in all these positions. So from the healthcare, it's really important to monitor what mutations are there, because it's like with a flu, you have to create a new vaccine each year, because each year there is a different strain of flu. Here, if we are going to treat coronavirus with different monoclonal antibodies, we have to create new one in advance to see to be prepared for the each year wave which we can expect. And this is the difference between all variants which are important here in Europe. So this is the original one with mutation DAUG here, alpha, beta, gamma, delta, and you see Omicron, how many mutations are there, and all of these mutations are exactly on the position where those antibodies binds. So if you have antibodies against the variant Delta, they will not work against variant Omicron, and we see it in a population that it's easy to get the coronavirus infection second time, because just our antibodies do not work much. And if you will have antibodies against that particular Omicron, they will not work very good for example, variant Delta. And now we are in the stage that we have now two different types of coronavirus strains. One are all those alpha, beta, gamma, delta, which are all similar, and basically antibodies works. Yeah, sometimes one of the type of the antibody from the alpha does not work against delta, but in general, most of them work. So that is epidemiology was really close. But now we have the Omicron, which is totally different. It's in a different place of that tree. Okay, so the reason why the Omicron wave was so strong was not only that it was able to spread faster, but also that the level of antibodies again that was really low again in the population, and basically it has against free place to expand. And now, what will happen next? What we can expect? Let's look how it is with other viruses of the similar nature. So these are influenza, which is monitored quite well. And what you can see here is that this is an evolutionary tree of influenza A, H3 and 2. And what you see is that it slowly changes, the mutations changes its surface, and our immunity tries to follow it, and then the overall evolution trait is that it just slowly moves together with us, and then nothing happened. Each year we have a wave of the influenza A, and each year we have to develop new kind of antibodies. Okay, that would be really great if this happened with coronavirus. Then influenza B. There's a little bit different situation, because we have two lineages, Yamagata and Victoria, and each year there is one of those variants dominant in the population. Here you can see it, and this is the whole world. Here you see the waves of the influenza during the year before the pandemic, and the strains, and you see that influenza B 1 year, and then influenza A, and then again influenza B. Now you see how our fight against the coronavirus was really great, how the influenza stopped we can get rid of influenza if we would like to. That's possible. You can see. And in 2021, there was no influenza at all. And probably Yamagata lineage was extinct. We do not have any sample from the whole Europe for the Yamagata influenza B strain. And here you see this is one region, how the different strains dominate. That's a really great way from the viral point of view, how to deal with our immunity, okay? Because you have two distinct strains. One is growing 1 year, the population develops the antibodies against that particular strain, okay? And next year you have a different strain. Antibodies are not there, so you have to develop antibodies against another strain, and then next year the immunity then wanes, and then next year the original one again starts to grow. Okay, so we can see it also the situation with coronaviruses. These are those two coronaviruses which are normal, OC43, 229E. 229E, it behaves like influenza A. It's just changing through the time. OC43, there are two different strains and they are probably changing each year. We do not have much data from there because coronaviruses were not studied much before the pandemic, okay? So that's the way, what can happen now next. Yeah, and that's again with the antibodies, you see that antibodies, okay, on this picture, you see either people who were infected by different strains or people with different vaccines, and neutralization by Omicron of different sera, and you see that all those antibodies basically does not work against Omicron, except really fresh vaccination or combination of vaccination and disease. And yeah, it does not end here. We already see new strains which starts to overgrow the variant Omicron. Well, these are still Omicron, but we will see. This is the situation in New York and regions around the New York. This is Omicron, and now last week the variant BA2 start to grow there, and even before it were able to overgrow it, another variant BA2.12.1 starts to grow there. And each of these variants is again worse than was the variant before. It grows faster. It's more dangerous than was the variant before. This is New York. Here is the situation in South Africa, where we see two different BA4 and BA5 variants starting to grow there and overgrowing the BA2, which was presented there before. So this is definitely not end of the pandemic. The virus still evolves, and the situation between the new virus and new host is still not stable in a way like it is with influenza. Still, there is a lot of place for the virus, how to change and how to change its behavior, how to change its infection potential, and how to spread through the population. And of course, the situation is even worse because RNA viruses are known that they can easily recombine, and that's what we see. Of course, we think that there was a lot of recombination during the last 2 years, but we didn't see them because there was not enough mutations. But it is now, so we can detect number of different recombinants between different variants of the strain. So here it is BA1, BA2, a number of them were found. And also we have recombinant between Delta and BA1. From the epidemiological point of view, that's a problem because you have a variant, for example, this particular XD from France, which has spike protein from the variant BA1, which allows really fast spread, and the rest of the genome is from Delta, which actually was quite bad from the health point of view. It killed a lot of people. So this is like accelerated evolution. You can choose two different parts of the virus, one which is good for the spread, the other one which is bad for us from the health point of view, and create it. Fortunately, none of these recombinants, you see that recombinants, the name convention of the recombinants is that they started with X. So none of these recombinants start to grow really fast. but we will see. Okay? So what can happen next? There are three possible scenarios which we can take into account. First, hopefully that will be the end of the pandemic, is when we become compatible with the virus, and the virus will slowly change its genetic information, and our immunity will slowly follow, something like influenza A model. We can have the other one, which is much worse. Two strains, for example, Omicron and Delta, will be circulating in the population, and each autumn we will have to observe which one is dominant then, and change the treatment according to the variant which will be there. And of course, we are still not at the end. Still there can be totally new variant with totally different serotype, like it was with Omicron, where no antibodies which we have now will work really good. Of course, they are working somehow. There will be no really bad. Well, we think that the vaccination, or we have data for that, the vaccination works really good against the death and against the hospitalizations. So it probably will stay, but against the infection, it does not work much. And if we will have a new variant in the autumn, then we can see again a really high number of newly infected people again and again. What will happen, nobody knows. But these are three scenarios which are now be taken into account. Okay. So there is a lot of we can talk about. If you would like to, I'll be here around, so you can catch me and we can talk about the details. And thank you for your attention, and give me your answers, your questions.
Okay, so time for discussion. Please, is there anyone who's there to ask us? Yeah.
Musíš mu dávat mikrofon.
When you call the variant dangerous, what does it mean?
Really good question. That's something which is relative, depending on the situation which is now in the particular state. There are some variants which made a lot of dead people in one state and not so much in other one, and vice versa. So it's really hard to say. But in general, what I'm talking mostly is the genomics. So it's a dangerous in a way how fast it can spread through the population.
It's not a follow-up question, but a second question, which just might be stupid. Why the winter, summer variants? Why does the virus go down during the summers?
That's common in all respiratory viruses, because the weather they does not like heat and
Where does the virus go during the summer?
Oh, there are that's really great question. There still are people who are catching the virus even during the summer. And for example, in places where there is a really hot, then the situation is reversed, because the people are gathering together when there is a hot outside, and they are going to the buildings together with a crowd, with air condition. So, for example, in South Africa, they will have the wave during the summer, not during the winter, right?
More people outside,
Better, well, worse for the virus. It cannot spread so easily. It does not like hot weather. It does not like really cold weather. Humidity has to be so, but that's exactly the condition. You see, the wave starts when the heating season starts, because at that time, the humidity in the buildings drops down dramatically from 60% to 40%. It means burden of our sliznice. And then it you can catch it much easier. So it's true for all respiratory viruses, influenza, RSV, whatever.
Paulina, sorry.
Thank you for your talk. So now all the changes and mutation were response to evolutionary pressure of antibodies, immune system, and so on. Now we will have some virotics or antibiotics, hopefully. So can you somehow extrapolate or guess how the virus will respond to this and how fast the resistance will evolve?
Yeah, that's what people are quite afraid. There is a number of different antibiotics. So you have those like molnupiravir or something, which makes it difficult for virus to reproduce because this is analog of RNA. It makes a lot of mutations inside. So if you will take the molnupiravir in low levels for a long time, that's a really nice tool how to create much more variations and much more mutations in the populations, right? But for example, Paxlovid, that's against the protease, and there should not be resistance against that. But actually, who knows? And we have to monitor it. If that's the reason why actually each country has to sequence quite heavily. Yeah, it's those 25 per year, and how many you will need for the resistance, that's really hard to say, because we understand that surface, because that's easy. It's antibody and binding to protein, but the resistance of the protease, for example, what the changes will be there, I don't think that nobody really knows the dynamics of that. How many mutations you will need to escape that particular small molecule. The fight against small molecule is much worse, much more complicated from the organism point of view, because it's not so complex like the whole antibody. For the antibody, there is enough one change and the antibody stopped work. Usually for the small molecule, it's not true, or there's only one position where can that particular mutation happen. So yeah, I believe that we have much more time to develop the resistance. Hopefully.
Great adventure for you.
Oh yeah, that is going to be great adventure, right?
Some comments.
Yeah, I wanted to ask Paulina's question, so but follow up actually. I mean, we do have like more than 30 years experience with resistance development towards small molecule, and unfortunately, I have to tell you, it will develop for sure. I mean, no question about it. With HIV protease, we spent years on spending of structures of resistance mutants towards HIV protease. The same is true for HCV. The same is true for laboratory strains of influenza. So it will develop. My question was whether or not you are ready to follow it, because it will be here. So again, are you ready to sequence the protease portion of the virus as well, and the check for the resistance development?
Yeah, I think that well, I personally am ready, right? But the thing is that, for example, from today or from yesterday, there is no one free test per month. It means that we will have no idea what variants are going to grow. And all those test sites, which took us 2 years to develop, now they will all be disbanded, and nobody will have power to start it again at the autumn. So for some countries, for example, I am quite afraid that the monitoring for the next year is not going to be sufficient. But hopefully the other countries like Denmark and UK will help us. That's the only what we can bet on.
And one remark, as the other people here, we would like to acknowledge your beautiful work during those 2 years. It was amazing and very important. Congratulations, or it goes to this group of people. That's wonderful bunch of people helping a lot. That's great. And actually, the reason why we were able to do that was also the ELIXIR, because there was the infrastructure which immediately gave us a computational power and place where we can store the data, and they allowed the other people to connect to that site and build it. And it was like this. We just asked about that. So
if there is big infrastructure asking for your support, do it, because without these large infrastructures and Elixir especially, you cannot do you cannot solve such a situation in normal time. That was great.
Just acknowledging that we have the national hub here at the institute, so we are very proud.
Yes.
My question is related to the phenomenon called virus interference. In some sense, what are the current mechanisms or proposed mechanisms how one variant displaces the other? Most recently it was like Delta, it was a peak of Delta, and Omicron started, and there was a very short period in between. So what is really happening? Is it, what comes to mind is this innate immune response, so virus which gets there first triggers something, and the second virus simply cannot catch on. But is there anything, or are there sort of concomitant infections where the viruses are really fighting in the same moment?
In fact, the reason why it is so fast is the exponential curve. It's enough to be a little bit faster than the other one, and during such a huge number of infections, you will just overgrow it very fast. If you calculate it, it's really 10% difference makes the exchange very fast, very.
If there is no interference, you can imagine that the two viruses will simply coexist. There will be a wave of Delta and a wave of Omicron, and simply they will overlap, and there will be no interference. But apparently there is.
Yeah, that's.
So what mechanistically or how does it really work?
That's the situation, I think, which is stable, and we think that it will happen. Why now it is not happening is because it is still, the virus still have too many possibilities how to be better. Because the situation between the new virus and new host is not stable from the genetical point of view. You need several years to develop that. And it happened with 1918 with the influenza, the same. The first 2 years was really bad, and now H1N1 is a bad influenza, but a normal situation. So probably the reason is that the contact between virus and the host on a surface level is still not optimized. I don't know about that particular enzyme, but for the respiratory viruses, it's not only the speed of the virus, but it's contact between people, which makes the epidemiology. And so the behavior of the people triggers those waves, not the virus itself. That's really great idea. I have no answer to that. But yeah, well, that's something we should think about. I didn't read any article addressing that, so I don't know. But it's really interesting. Yeah.
There was a question.
You mentioned that it's possible that more dangerous variant will develop. Why would evolution favor a more dangerous virus? Because we would react on it and we would close up the
evolution does have nothing to do with that. It just is a random process. And the virus can be really bad in terms of that it can destroy the organism. Sometimes it happens, sometimes it's not. And we see those situations where it does not happen. So who knows what happens? And so far, why it is still each strain or each variant is worse than the variant before in terms of the speed of the reproduction is because this optimization process. It was new virus, it was not optimized on our body and our immunity and our cell surface, and that goes in that direction. Mm-hmm.
Just one question, could you speak? Do you think that there are people who are genetically resistant to SARS-CoV-2?
Yes. They are, yeah. But, well, resistant not to infection, but we know that they already are known SNPs, single nucleotide polymorphism, which are connected with a bad progress of the disease or with a good progress of the disease, but not against infection. The Ac2 receptor does not have variant which will protect you against the binding of the virus so far.
Yeah, I probably question, did anyone try to infect a cell by 2 different strains and observe what is going to happen? That probably knows about it.
Yeah, that's the research which is not supposed to be done because it's dangerous, and information coming out of that research is not so important. So, yeah, well, you probably can think about that, but with the origin of that particular virus, there are some doubts how actually the virus jumped to the human, and now all this research. This is it.
What I meant, take Omicron or Delta, infect a cell and observe what is going on. Which of the variants will be the stronger? Is that possible, like in an experimental?
Oh yeah, definitely it's possible, but I didn't read any article.
Of course, but it doesn't tell you about the pathology in the whole organism. It's a completely different story.
On a cell culture,
Of course, we have data in cell cultures. I mean,
yeah.
you can derive some preliminary information about the pathogenicity of the particular strain, but to extrapolate for the whole organism, it's much more complex because it's the immune response of the organism that in fact kills the patient rather than the virus itself.
And the stage of the whole population from the immunological point of view is also which makes much more than the virus itself.
Okay, time for last 2 questions.
Can you just repeat the questions for the people who are watching on YouTube?
Question.
Yeah, because the questions are bad to hear.
Okay, yeah. Yeah, I will repeat the last question. Sorry, it's too late. I'm sorry. No, from now on. Okay. So there was a question, the last one.
Regarding the influenza B lineage going extinct, how would you explain that? Could it have been caused by safety measures, or was it overnight somehow?
Okay. Question is why one of the influenza B strains is extinct. Yes, the measures we take as a whole world take against the coronavirus are the same, because it's respiratory virus like against the influenza, and on that particular curve you can see that basically we stop the spreading of the influenza on the level that one of the strain probably was extinct. During the summer there was not enough people infected and then it disappears. That can happen probably with all other viruses as well, if whole world will fight at least 1 month against it without any error. Which will never happen, of course.
So sorry, but in that case, would it be smarter to do all the restriction during the summer?
First, we do not have the same summer and the whole earth, so that's one thing. And yes, it would be, but you see that different countries took different ways how to deal with that. Some of them worked very well at the beginning, where the virus was not so dangerous and not so fast. But now it does not work. This zero COVID strategy was probably a good idea 2 years ago, but not anymore. So yes, that's the time where we can get rid of the virus, the summer. Of course, not during the winter, but not anymore. But my opinion.
Just a remark, I would not forget about the animal hosts.
Yeah, that's one thing. The virus is strange. It's different from other respiratory viruses because it infects much more, many more tissues, but also it can easily jump to other species. So hamsters, for example, the wave in Hong Kong was started by hamsters, actually probably from the Czech Republic, or the closest sequenced variants were from the Czech Republic. So you can be infected, you can infect hamsters and they can infect you. Hamsters are spreading the virus even better than human. There was a big outbreak of minks in farms. They were killed away like crazy in Denmark and so on. In North America, the white-tailed deer, 80% of them are infected by the coronavirus. Nobody knows why. It's well, in farm, of course, if there is a man who is infected and taking care about the animals, you will infect them. That's clear. But white-tailed deer, what those hunters are doing with them, I really wonder. 80%, there are some regions where 80% of white-tailed deer had coronavirus, the SARS-CoV-2 virus. So yeah, that's another way that now we cannot get rid of them, because it's not only in humans, it's probably in rats as well. And there is one hypothesis how Omicron developed. One is that it was in immunocompromised person, and the other one that it was from the host, that it jumps from the human to probably rat or horse or whatever, and then after months it jumps back, and that was the reason why it has so many different mutations than the other variants. But that's a hypothesis, not easy to prove it.
These evolutionary pressures. So question is about some of those remedies we are taking, like vaccination or face masks, whether they contribute to basically selecting variants which can avoid them. And I was actually asking that question in Gilead. There was a virologist and so on, but the answer I got sounded too politically correct. They kind of, no, those don't actually, they have nothing to do with selection of those new variants, because the new variants actually arose, some like in India, the Delta, or in South Africa, or something in Brazil. And these are countries where you don't have those measures, actually vaccination level is quite low. So in other words, those things don't contribute to selection of those new variants. But evolutionary speaking, it doesn't make sense, because I mean, clearly you generate mutations and then you select, and whatever the obstacle is, so you select the variant. So what's the idea?
Okay, vaccination is no different from the normal infection from the evolutionary point of view. You just have antibodies against that, and of course you see that if everyone here has antibodies against the Omicron, then the variant which changes those epitopes will grow easily here, because we will not have variants against them. So in that term, infection and vaccination can contribute in the equal level, and you can do anything against that. The face masks and other behavior, it's really hard to imagine how that can change it. Yeah, there are people which they have behavior which is more favorable for the spreading of the virus. People who are using masks or people who are not using masks, these are different kind of people probably from the behavioral point of view. And then if there is some genetics behind that may be. But in fact, I cannot imagine the procedure how that, for example, mask, I don't know how that, the virus will not change the size, that's for sure.
Okay, so probably the last question. I see Milan. I see you. Now a question.
Could you go to one of the first slides with the life cycle?
Whoa, so many slides. Sorry for that.
Even before, yes, there is this inhibitory effect of hydroxychloroquine.
Yeah, that was the reason why, this is one of the first articles, and that was the reason why they start to test it, because it has, the idea was that it can bloom changing the pH, so there was a rationale behind that experiment. But it didn't work, because the main way how to get in is this one, and that's the reason. But Omicron changed that, because Omicron now not prefers, but is using the endosomal entry much frequently than the other one.
Donald Trump was right.
Okay. No. I mean, there was a lot of attempts to find a treatment for that, and all of them had some generally good idea behind that. That's the normal way, and then you test it, and in some of them you see a difference when you are using the treatment. And some of these they didn't work. And many other molecules were used for as a test for that. Ivermectin is another one. Yeah. But now we have really big studies to show that they do not help.
Okay, I'd like to thank Honza again.